All right, so I have now completed Chapter 1 of the Biology Review book for my 7-book complete MCAT review and preparation. And Chapter 1 consists of 1.1, Cell Theory, 1.2, Eukaryotic Cells, 1.3, Classification and Structure of Prokaryotic Cells, 1.4, Genetics and Growth of Prokaryotic Cells, and 1.5, Viruses and Subviral Particles, and then a concept review. So, 1.5, we start off with cell theory. So, the 4 basic tenets of cell theory are that all living things are made out of cells. Cells only arise from other cells. A cell is the most basic unit of life. And a 4th one that was recently added upon discovery of DNA and everything is that DNA passes on genetic material from parent cells to daughter cells. 1.2, eukaryotic cells. So the learning goals for this section are: explain the importance of hydrogen peroxide to cellular function. Number 1, hydrogen peroxide is housed in peroxisomes and helps break down very long-chain fatty acids. Learning objective number 2, identify the predominant proteins found in microfilaments, which is actin, microtubules, which is tubulin, and intermediate filaments, which is like keratin and a couple of other ones. So yeah, okay, section 1.3, yeah, this is a long one. Yep, so you got keratin, desmin, vimentin, and lamicin. Lamins, the last one. So, section 1.3, classification of structures of prokaryotic cells. Compare and contrast archaea, bacteria, and eukaryotes. So, I guess the best way I can think of to compare and contrast archaea, bacteria, and eukaryotes is to say that archaea are similar, don't have any membrane-bound organelles. They are similar to prokaryotes, meaning bacteria, in visually, but their cellular processes are more akin to eukaryotes. Bacteria are obviously prokaryotes. They don't have membrane-bound organelles. They synthesize ATP very differently than eukaryotes do. They use a cell membrane instead of mitochondria, of course, to create the gradient for proton transfer. And eukaryotes, obviously, cells that make up humans and animals and everything, they have membrane-bound organelles and are much more complex and much larger than prokaryotic cells. So identify the 3 common bacterial shapes. You have rods, you have spherical, rods, and spiral shaped bacteria. Spherical is Kochi, rods is Bacilli, and spiral is Spirilli, excuse me. So explain the difference between gram-positive and gram-negative bacteria. It's the 3rd learning objective for 1.3. So gram-positive bacteria, gram-positive versus gram-negative refers to when you are staining the bacteria with different stains. So there's one particular stain that you will use. The name of it is escaping me, but that's not particularly important to this anyway. The gram-positive bacteria will appear as dark violet, like a very deep purple color, and that means that they have a much thicker cell wall that contains a much thicker cell wall that contains much more peptoglycans. Yeah. So it absorbs the stain and appears as a deep purple and has a thick layer of peptoglycans. And gram-negative absorbs the counter stain and appear pink, like a light pink color. And they have a thin layer of peptoglycans. So detail the structural differences between eukaryotic and prokaryotic flagella. So eukaryotic flagella are composed of basically entirely tubulin and a membrane. So they have microtubules that run through them in a 9 and 2 structure. So that means that they have 9 tubes running all around the outside of them, and then they have 2 on the inside, and that gives them their shape, and then that's all surrounded by a by a membrane, of course. And then prokaryotic flagella are they have several different sections, and their actual, I believe it's called excuse me, one second. Their filament, yes. Their filament is hollow. It's also made of microtubules, but it has the 9 going around the outside, but it's hollow. And then they also have a thing called a hook and L-ring, rod, P-ring, and they're composed of very different proteins than a eukaryotic cell, eukaryotic flagella would be. Differentiate between the metabolic processes of aerobic and anaerobic bacteria. So aerobic bacteria, they can use oxygen to produce energy. Anaerobic bacteria cannot use oxygen to produce energy, meaning ATP. So we have 4 different types of aerobic and anaerobic bacteria. You have obligate anaerobes, facultative anaerobes, obligate aerobes, aerobic bacteria, facultative anaerobes, obligate anaerobes, and aerotolerant anaerobes. So obligate aerobes, right? So they can only survive in the presence of oxygen. Anything else, they die. So facultative anaerobes, they are able to survive with anything. They can survive with oxygen or without oxygen. Obligate anaerobes, meaning that they can only use other things. They can't use oxygen to produce energy, and they will die in the presence of oxygen. Aerotolerant anaerobes mean that they can survive with the presence of oxygen, but they can't use it. So yeah, that is 1.3. 1.4, describe bacterial genetic recombination via transformation, conjunction, and transduction. So bacterial genetic recombination via transformation. Transformation is when a bacterium incorporates outside genetic material that it has endocytosed into itself, often incorporating different plasmids into its own genetic DNA. It's basically taking outside genetic material, bringing it into its actual genetic code for replication. Conjunction. Conjunction for bacteriums is basically sexual reproduction for bacteria. There is a bridge between 2 bacteriums that is formed, made of proteins, I guess, and there is genetic material that is fed through that bridge, and there's a whole bunch of things that you need to know about, fertility factors and other things, but that's not particularly important right now. Obviously, we'll be looking back over this later. And transduction is genetic recombination facultated by a viral vector, which means that you can only this method happens when there is a bacterium, and that bacterium gets infected by a virus, and then the virus, in recombining, somehow pieces of that bacterium's genetic code gets into the capsid of the virus, and so then when that virus goes on and infects other bacteriums, that piece of genetic material from the original bacterium gets injected or endocytosed into the other cell and incorporated just like the viral DNA is. Recall the 4 phases of the bacterial growth curve and the major features of each phase. So, yes, there are 4 features. Okay. Let me read. Okay, yes. So the lag phase, it's the first one. Couldn't remember that name. So the lag phase is basically the bacterium adjusting to its environment. It is the bacterium weeding out all the bacteria that aren't suited to reproduce, and once the lag phase is over, you hit the exponential phase, which means that the bacterium is growing at an exponential rate. This is what you would think of when you see examples where you're like, a bacterium replicates every 30 minutes. How long will it be until the bacterium is at this many bacteria? That's always going to be an exponential phase. Once you hit the stationary phase, that means that the environment has reached its carrying capacity for the bacterium, and all the food sources that are in that environment are being used up. And once they are all gone, you hit the death phase, which the curve drops back down, and all the bacterium are dying because all the food is gone, basically. Okay, 1.5. Explain why viruses are defined as obligate intercellular parasites. So viruses are defined as obligate intercellular parasites because they are incapable of reproduction outside of a host cell, meaning that the virus needs to utilize the mechanisms and structures within a host cell to produce more viruses. So they cannot reproduce without that assistance. Recall key viral technology, including retroviruses and positive/negative sense. Virus terminology, excuse me. So retroviruses basically means that when the virus infects the cell, the viral DNA is directly incorporated into that cell's genome. So that virus will continue to be replicated as that cell is dividing. So HIV is a retrovirus. It is extremely hard to treat retroviruses because the only way to stop that cell from reproducing that virus is to kill the cell. And there's not any other way to treat it because that cell has now been tainted by the retrovirus because its genome has been taken over. Positive or negative sense. So positive sense means that when the virus injects its genetic material, it can be immediately directly encoded into functional proteins to make more viruses. Negative sense means that the protein that's injected into the cell injected or endocytosed into the cell must first be coded into a different form. It must be coded into a complementary strain, which then can be turned into functional proteins. Describe how viruses are able to produce progeny via infection of a host cell. Kind of went over that a little bit earlier. So viruses inject their genetic material or are endocytosed through the plasma membrane, and they will, depending on the structure of the genome that is within the virus, they will enter the nucleus or go directly to the ribosomes for protein synthesis. Assuming that the virus has 2 double-stranded DNA as their genome, they will need to go into the nucleus so that they can use RNA polymerase to split apart their strands of DNA and then turn that into mRNA before they can actually protein synthesize. But if they have single-stranded DNA or single-stranded RNA, they can put that directly into the ribosomes and it will be coded into proteins. And then, of course, once proteins are being coded, then the viruses are assembled and then they will extrude from the cell, or they will build up for long enough until the cell lyses and dies and they all just escape. Compare and contrast lytic and lysogenic cycles. So The lytic cycle basically means that the virus has no regard for the survival of the host cell. It will use up resources until the host cell dies, and then the cell will lyse, and all of those viruses will basically explode out at one point. The lysogenic cycle means that the virus incorporates its DNA into the host cell's DNA and replicates as that host cell replicates. And this will not kill the cell, and the viruses that are being replicated are extruding out of the cell, meaning that it does not kill the cell for them to leave. When a cell is in the lytic cycle and they are infected with a virus, that virus is referred to as virulent, and in the lysogenic cycle, that is referred to as a productive cycle. So describe how prions and viroids are able to cause damage. So prions are basically malformed proteins that can enter into your brain if you're a human, and basically they will cause proteins that are correctly formed in a alpha helix to be misshapen into a beta-plated sheet, and they're not alive, just like viruses are not alive, but they still cause damage by misshapen proteins, and when those proteins build up, they can block off things and cause lots of problems. Viroids are very small. Viroids are very small pathogens consisting of very short circular single-stranded RNA. So they're more common in plants, but they do exist in humans and animals. So some of the more well-known viroid infections are hepatitis D and HDV, and hepatitis B. So that's chapter 1 of the biology review for my MCAT. Yay. Thank you for watching. Bye-bye.